All right. So I think maybe now is a good time to start. So thank you everyone for joining us today for another exciting Rapid Novo webinar. My name is Wei, and I will be moderating today's presentation. Today's webinar is titled Maximizing Liquid Biopsy Sensitivity Through the Power of DNA Binding Antibodies. But before we begin, I'll just go over a few housekeeping tips. First of all, we will be doing a Q&A at the end of the session. So if you have any questions, please type it into the Q&A chat below. As well, the presentation is being recorded and the recording will be made available to everyone afterwards. All right, so introductions, if you could please advance the slides. So today's presentation will be given by our guest speaker, Dr. Shervin Tabrizi, and our very own Jennifer Surha. Dr. Shervin Tabrizi is a physician scientist at the Department of Radiation Oncology at Massachusetts General Hospital and Harvard Medical School, and a researcher at the Broad Institute of MIT and Harvard and the Koch Institute for Integrative Cancer Research at MIT. His research focuses on liquid biopsies and combines approaches in genomics, protein engineering, cancer biology to develop novel technologies for cancer detection. So thank you so much for being with us today. Jennifer Surha is our regional sales manager and is a dynamic professional with experience in scientific sales and research, the biotechnology and life sciences industry. She's currently serving as a regional sales manager at Rapid Novo, and Jen specializes in consultative sales for antibody sequencing, discovery, and characterization. So I will now pass it over to Dr. Tabrizi to begin the presentation. Thank you for your attention.
Thank you so much for that kind introduction. It's a pleasure to be here with you this morning. So I'm going to talk to you today about some new technologies we've developed to try and get over one of the big limitations in current liquid biopsy technologies. So to start, liquid biopsies have really garnered a lot of attention recently because they can enable cancer detection from a blood draw, and often they can do so before the cancer becomes evident on imaging scans. Among all the liquid biopsy technologies, the technology that's really gotten the most traction in clinic, at least for cancer detection, is cell-free DNA liquid biopsies. And specifically here, what we're talking about is the detection of tumor DNA fragments within the circulating cell-free DNA component in plasma. The idea is that all the cells in the body, including our normal cells as well as tumor cells, shed their DNA into circulation, and this DNA can be recovered from the cell-free or plasma fraction of blood. And so when we get a blood sample from a patient, we can recover both the normal DNA here in gray, but also some fragments of tumor DNA. And we can use this as essentially a biomarker for detecting the presence of cancer in the body. Now, there's been a lot of excitement for using circulating tumor DNA as a biomarker in oncology. And a lot of it's driven by data like this, where you can see that circulating tumor DNA really has exquisite specificity for detecting patients who are going to recur pretty quickly, as you can see by the separation in the curves between ctDNA negative and ctDNA positive patients. And this has been recapitulated in many clinical studies. And so because of this, this technology is now being explored as a tool across the spectrum of cancer care, from early cancer detection to detection of recurrence to even monitoring and selection of therapies for patients with advanced cancers. But there's still a major problem with applying this technology in the clinic. And the problem is that the sensitivity of current tests is still quite low. So these tests are very specific. If they're positive, the patients are going to recur quite quickly. But the problem is that in most applications, they still miss a vast majority of patients who have cancer in the body. So in early detection, the sensitivities that have been reported are somewhere in the 20% range for detection of recurrence, maybe a little bit better, but still 25 to 50%. And even for patients with advanced cancer, where they have the highest level of tumor DNA in the body or in the plasma, the sensitivity can be still in the 25 to 50% range, especially when the tumor DNA content is below about 1%. And so this is a major limitation if we want to actually use these technologies in the clinic, because what it means is that a negative test is hard to interpret. We don't know if it's negative because the patient truly doesn't have disease or because the test is just not sensitive enough to detect the presence of cancer. So we wanted to address this problem and to understand why this problem occurs, we can turn back to the biology of cell-free DNA and circulating tumor DNA. It turns out that when there's a lot of tumor in the body, there's also a lot of circulating tumor DNA in plasma. But when the tumor burden is low, those circulating tumor DNA fragments also become much less common. And in these situations, when we take a blood sample from a patient, we can often miss those rare tumor DNA fragments in our blood sample. And here, when the tumor DNA is physically absent in the blood sample, there's no way to recover that signal after you've collected your sample. No matter how deeply you sequence, how accurate your sequencing is, you're just not going to be able to detect tumor DNA because it's physically absent. And so this is a limitation that's separate from technical limitations regarding like sequencing noise and things downstream, in which there's been a lot of advances over the years. This is a fundamental limitation with the sample itself. And so we thought this is really the bottleneck that we need to address if we want to overcome the current sensitivity barriers for liquid biopsies. Now, it turns out that one idea that we could think about is just increasing the total concentration of cell-free DNA in plasma. This is something that sort of analogous to what a lot of liquid biopsy companies even today require, which is taking multiple tubes of blood. Basically, you're increasing the total mass of cell-free DNA you have. And what it does is it basically increases the chance that you will pick up those rare tumor DNA fragments in your blood sample. And so, one thing, what we thought is perhaps if we have a way of actually increasing the concentration of cell-free DNA in plasma, we could use this as a way to overcome this sensitivity barrier. The idea of collecting a larger volume of blood, although it seems pretty straightforward, there's a very obvious limitation in that at some points and pretty quickly, your test no longer becomes non-invasive and could actually be, there's practical limitations to how much blood you can collect from a patient. So we thought to really break through this barrier, we need to come up with an alternative strategy to improve the recovery of cell-free DNA from a blood sample. Now, it turns out that cell-free DNA is degraded through various mechanisms. The major ones are through uptake by organs like the liver, as well as degradation by nucleases that are in circulation and in tissues. And one way we can think about increasing the concentration of cell-free DNA is by actually blocking these pathways. And in that way, you can allow cell-free DNA, both normal and tumor DNA, to build up within the body. And so then when you get a blood sample, you have a much better chance of picking up those rare tumor DNA fragments. And what we thought is, well, what if to accomplish this, we could actually couple cell-free DNA to a protective molecule that would basically shield it from clearance via the liver and other organs and degradation by nucleases. And for this purpose, we decided to focus on antibodies for a number of reasons. They're easy to engineer. They have known pharmacokinetics and ways of manipulating those pharmacokinetics. And they also have a long track record as biopharmaceuticals. So that was the impetus for this project. And as a first step, we needed to just pick an antibody that could bind cell-free DNA. And we tested a panel of 8 known antibodies, and 4 of them showed on ELISA some binding activity to double-stranded DNA. Of those, we picked the one that was best characterized in the literature, this antibody called 35I9, which is an IgG2A isotype antibody with a KD of 90 nanomolar to double-stranded DNA and 700 nanomolar to single-stranded DNA. So relatively specific binding to double-stranded DNA. We first wanted to see if this antibody actually bound cell-free DNA. So that's both free DNA fragments as well as DNA that's wrapped around the histone octamer, which is thought to be how most cell-free DNA exists in circulation. So we did just a gel shift assay. Here, I'm showing both free DNA and histone-bound DNA. And we saw that with increasing concentrations of the antibody, we actually saw these shifted bands corresponding to the binding interaction of the antibody with both free and histone-bound DNA. And we saw the disappearance of both the free and histone-bound DNA, suggesting that this antibody could interact with both components of cell-free DNA. We further verified that it was actually interacting with histone-bound DNA by just doing an immunoblot for histone H3 on the same gel and saw the same shifted bands. So this is consistent with the antibody actually interacting with mononucleosomes. We also then tested the ability of this antibody to in vitro to protect DNA from nucleus digestion. So we used an assay of basically a DNA substrate with a fluorophore and a quencher, where DNA's degradation leads to a fluorescent signal. And we saw that with higher concentrations of the antibody, we got decreased fluorescence consistent with protection of the substrate from DNA's digestion via the antibody. And similarly, this is for free DNA. We did the same experiment then with mononucleosomes. And again, we saw that some protection of the mononucleosome-bound DNA in the presence of DNase with the antibody. So this suggests that this DNA-binding antibody can actually protect both mononucleosomal and free DNA from degradation in vitro. So next, we wanted to see how does this actually affect the clearance of cell-free DNA from plasma. So to do this, we injected mice with this antibody plus mononucleosomes of a known sequence, and then we measured the concentration of the DNA sequence that we injected, called WDM601, via qPCR over time. And what we found is that when you normalize to the initial sort of 1-minute levels of WDM601, then in the presence of this DNA binding antibody, either 10 micrograms or 40 micrograms, you actually see decreased clearance of the injected WDM601 sequence. So this tells you that the clearance rate of DNA is slower in the presence of this antibody compared to an isotype control or compared to just the without antibody. And so this is what we're looking for. We're basically looking for a way to slow down the clearance of cell-free DNA from plasma. But unfortunately, when we look at just the absolute levels of WDM601, so this time we're not normalizing to the 1-minute mark, we actually didn't see a difference at 60 minutes. And this is because we actually saw rapid clearance of a large fragment, a large fraction of the injected DNA at the 1 minute mark. And this seemed to correlate with the dose of antibody that was injected, DNA binding antibody that was injected. What we hypothesize here is that perhaps the antibody is interacting with Fc receptors, FcγR receptors, and sort of sequestering and leading to rapid clearance of a large fraction of the DNA that we're injecting. And so to do that, to test that hypothesis, we did the same experiment, but this time we first blocked FcγR receptors in the mouse with anti-FcγR receptor antibodies. And we saw that indeed we could actually rescue this effect. So this tells you that FcγR receptors would play a role in clearing these DNA bound to antibody, and something that we want to avoid if we want to develop an agent that can improve the recovery of cell-free DNA. So we were able to do this with just blocking Fc receptors, but what we really wanted to do is to actually engineer the Fc domain of the same antibody to basically recapitulate this effect. Now, we did not have the sequence of this antibody. It had not been characterized. And so we then performed de novo antibody sequencing of the full heavy and light chain. And once we had the sequence, we were then able to actually do engineering of the Fc domain of this antibody. And we generated three new variants, which I'm calling AST2, AST3, and AST5, with different mutations in the Fc domain that should disrupt interactions with FcγR receptors. We then performed the same experiment, but now with these engineered variants, where we injected mononucleosomes with the antibody and then tracked the levels of the injected DNA in the mononucleosomes over time. What we saw was rapid clearance of the injected DNA with an isotype control, and decreased clearance in the presence of FcγR receptor blockade with the unmodified antibody. And then when we looked at the three engineered variants, we saw that one of them, AST3, actually recapitulated the effect that we saw with FcγR receptor blockade and actually exceeded it. So we saw, certainly over 100-fold improvement in recovery of the WDM601 sequence at 60 minutes compared to isotype control, and about 10-fold higher compared to the wild-type antibody with FcγR receptor blockade. So AST3 became the sort of engineered antibody variant that then we took further for additional testing. So up to this point, all the data that I've shown you relied on this somewhat contrived system of injecting an exogenous DNA sequence and then seeing its clearance over time. What we really want to see is, can this agent actually improve the detection of circulating tumor DNA that's endogenously released from a tumor in the mouse? So to do this, we turned to this MC26 lung metastasis model. So these are mice with MC26 cancer cell line tumors in their lungs. And the experiment we did was we took blood from these mice before injection of antibody. Then we injected either this AST3 engineered antibody or an IgG2A control antibody. And then we collected blood again from these mice 2 hours after injection. And then we had this developed this bespoke circulating tumor DNA assay specific for this MC26 cancer model to detect, measure the levels of circulating tumor DNA from these tumors in the plasma of these mice. And what we saw was quite striking. So with the AST3 antibody, we were able to see up to nearly a 20-fold improvement in the recovery of tumor DNA in the plasma of these mice. And so this order of magnitude, or over an order of magnitude improvement, is something that really isn't feasible with just larger blood volumes, but it is possible now with this agent to collect basically the same volume of blood, but get nearly 20-fold more tumor DNA. So we are very excited about this. But what we also wanted to know is, so 20-fold more tumor DNA, does that also get you more information about the tumor itself, the molecular profile of the tumor? So to do that, we looked at our data in a different way. So we had a test that could detect about 1,800 unique mutations in the tumors in the plasma of these mice. And here I'm just showing the results from the control group. Each column here is a separate mutation, and basically it's colored red if the mutation was detected in that mouse, and white if it was not detected in that mouse. And each row is a separate mouse. And on the right, I'm just showing you the total number of mutations out of these 1,800 that we're tracking that we were able to actually detect in the plasma of these mice. And you can see that in the control group, we actually missed the vast majority of these 1,800 mutations. The most we were able to detect is around 450. But with the AST3 antibody, we got a much fuller picture of the molecular profile of these tumors. And and we were able to detect certainly over half of the mutations, and I think on average around 75 to 80% of all the mutations that we're tracking. So this significantly improves our ability to not only detect tumor DNA, but to even detect specific and and potentially informative mutations. Next, we wanted to so what I've shown you so far is better molecular profiling of these tumors with this new agent AST3, as well as detection of more tumor DNA. But what we also wanted to then see is what's the effect of this on sensitivity. So on the sensitivity of your circulating tumor DNA test to actually detect cancer. So to do this, we performed some computational downsampling approaches to see what the effect would be across different numbers of mutations detected, or different numbers of mutations tracked. So different panel sizes, and you can see that across the board, the AST3 group shows much higher sensitivity for detecting circulating tumor DNA across a wide range of panel sizes. But then more importantly, we actually did the same, a similar downsampling approach, but this time for tumor fraction. So this is the fractional tumor DNA content in the plasma of these mice. And what we found is that, again, across a wide range of different tumor fractions, the AST3 antibody provided a significant boost in the sensitivity of our assay for actually detecting tumor DNA. And so what I've shown you is that this engineered DNA binding antibody gets us, orders of magnitude more tumor DNA from the same blood sample. It enables much fuller molecular profiling of tumors from plasma, and it shows higher sensitivity. It improves the sensitivity of our circulating tumor DNA tests. Now, all these results have been published earlier this year. In the publication, we actually described two approaches. So one is the antibody approach that I spearheaded and described to you. We actually also have a complementary nanoparticle approach that similarly helps us improve tumor detection. So here, the idea is that the nanoparticles occupy cells that normally take up cell-free DNA, and so allow for more of the cell-free DNA to stay in circulation. And what we showed is that with these nanoparticles, we can similarly detect more tumor DNA in plasma. The one downside of the nanoparticles that the antibodies do not have is they do dilute the tumor fraction, so the tumor content in the body. So it seems that the wild type DNA becomes more prevalent as compared to the tumor DNA when we use these, even though the absolute quantity of both tumor DNA and normal cell-free DNA increases. But similarly to what I showed, these nanoparticles also did boost the sensitivity of our circulating tumor DNA assays for detecting tumors with low tumor burden, or detecting tumors in mice with low tumor burden, and across a wide range of different detection thresholds. And so to just sum up everything, what we've shown is that an engineered DNA binding antibody, it can increase the half-life of cell-free DNA in plasma. It can protect cell-free DNA from degradation and from nucleus degradation, and it can boost the recovery of circulating tumor DNA by over 10-fold. And what we've seen is around 20-fold is what we can get with this technology. We've also, in this paper, shown that a nanoparticle agent can improve the recovery of cell-free DNA. The one limitation there that we're working on is the dilution in tumor fraction that we see with the nanoparticles that we haven't seen, fortunately, with the antibody-based approach. And so in these studies, we've demonstrated the promise of this approach in preclinical models, both to recover more tumor DNA and to enable better molecular profiling of tumors from cell-free DNA, and ultimately to boost the sensitivity of circulating tumor DNA tests. And what we envision as the ultimate sort of clinical application for these agents is that, the current status quo is that without these, and we call them priming agents, when we take a blood sample, a lot of times we have little to no circulating tumor DNA, which leads to low sensitivity and inconclusive tests. But that in the clinic, perhaps one day, patients can receive agents like this, and then when we collect their blood an hour or two after administering their agents, we can get significantly more circulating tumor DNA, and so have a much more sensitive test and have confidence in the results of those tests. Now, this is all, of course, preclinical at this stage. So the next questions for us are, how can we improve on this effect? Can we go beyond 20-fold improvement? Would it be possible to enrich specific cell-free DNA fragments, like tumor-derived fragments? So basically, in effect, almost boost the tumor fraction in the sample compared to what's found in plasma. And then ultimately, what are the challenges and questions that we need to address as we work on translating this to the clinic. So questions regarding dosing, tolerability, and of course, cost are going to be critical for ultimately getting this to the clinic and to the benefit of cancer patients. And so with that, I'll pause, and I think I'll hand it over to Jennifer.
Yeah, thank you for the fantastic presentation, Dr. Tabrizi. I'll share my screen now, and we'll get going with rapids. Okay, so in my half of the webinar today, I'll be going through how Rapid Novor does the de novo antibody sequencing that enables researchers like Dr. Tabrizi to obtain a sequence that is unknown and do some further engineering downstream. So a little bit of background on Rapid Novor. We are the largest privately funded proteomics facility in Canada. Really, that just means we have a lot of mass specs. That's all the work we do here. And so we've really hyper specialized in de novo sequencing of antibodies and proteins, as well as some other proteomics type of protein characterization services. We have around 70 employees, so we're not too big, not too small. We're able to pivot and do a lot of custom type of projects for people. And if a protein is particularly hard or difficult, we're able to spend more time on it and really work through any difficulties, and perhaps even do some like extra R&D that companies of a larger size might not be able to do. On the monoclonal sequencing side, we've sequenced over 9,000 antibodies so far. So yes, we do keep a tally of it. There are currently bets happening on when the 10,000th will arrive. And we are the first company in the world to be able to sequence polyclonal antibodies using only proteomics. So this is just a brief outline of our services. REMAB is the service name for the monoclonal protein sequencing that Dr. Tabrizi used. But just so you guys know, we also offer REPAB, which is called polyclonal sequencing. We offer a sequence confirmation service, and then some downstream characterization services such as epitope mapping using HDX-MS and antibody kinetics by SPR. So since we'll be focusing on REMAB, we'll get right into how we do that. So I like to break the MAB sequencing down into 4 steps. So step 1 would be enzymatic digestion, and then mass spec acquisition, de novo peptide sequencing and assembly, and then bioinformatics review and report generation. So going into a little bit more detail, of course, the first step is sample intake. There's a few things that we do on every sample that comes to the lab. So step 1 is just running a nice SDS page gel to double check the purity, make sure that there's not any contaminating proteins, and give us a general idea of what we're dealing with. So ideally we see 2 nice sharp bands for the heavy and light chain. That's not always the case. Sometimes we see gels like this that have a lot of BSA or other type of contaminating proteins, and so this is a really good first step for us to make sure that we're not loading a ton of BSA on the mass spec. So for a sample like this, we'd go and clean it up a little bit nicely, and then do the digestions on a cleaner sample. At this time, we also do what we call as a P-check, and that's a single pepsin digest. And with a pepsin digest, we're actually able to get about 90 to 95% sequence coverage of an antibody. And the P-check gives us a pretty good idea of, if the sample is going to be particularly difficult, or any kind of red flags that there might be before we do the rest of the digestions. That P-check also doubles as a quality control type of measure that we use later on, just to make sure that there's no sample mix-up in our lab, but also to make sure that the customer who sent the sample sent us the correct sample. So on another day, the rest of the antibody is digested, starting with 5 or 6 different enzymes. And those digestions are loaded onto the mass spec. We use Orbitraps in our lab because we need that really high sensitivity for de novo sequencing. And of course, the mass spec produces the spectral data, which is then converted into peptide data sets by our novo algorithm. And then basically like a puzzle, those peptides are assembled into the full-length protein sequence based on their overlapping amino acids. Of course, this is a really, really simplified version of what it looks like. And as you saw in Shervin's presentation, it looks more something like this, with hundreds of peptides overlapping. Each of these colored bars is a peptide, and the color of the bar represents what enzyme was used to obtain that peptide. And what we're looking for is hundreds of peptide overlaps here. And with that amount of abundance in peptides, that's how we're very, very sure of the sequences that we're giving out to people. What Rapid Novo tries to do is just give you a sequence that works. We don't want to mess around and have something that we're unsure of. What we're looking for is an overwhelming amount of data to give to people so that they can quickly move forward to their next step in their research. So, and of course, in science, there's always that one little curveball that it throws at you. And in the world of protein sequencing, it's the presence of isoleucine and leucine that have the same molecular weight. And so when people would be doing protein sequencing, 20, 25 years ago, you would have to rely on maybe the enzyme digestion rules or the germline sequence to guess if it's an I or an L. But what we've done is developed a patented side chain labeling method for isoleucine and leucine. And then we fragment those peptides further into the mass spec to obtain W ions. And from there, we're able to actually observe a mass difference. So leucine loses 43 Daltons and isoleucine will drop 29 Daltons. So we're able to see that on the spectral data and, with 100% confidence, know if it's an I or an L. In other parts of the sequence, there might be isobaric combinations, and our software is also able to tell the difference between those two. And so the last step of the process, like I said, is bioinformatics review. Of course, we're not going to give you a sequence that the computer just spits out and there you go. So we have at least two scientists reviewing every sequence that goes out. And this ensures that, the sequence meets our minimum quality standards and that there's no gaps or red flags in the sequence. And with us, the price you pay is the price that it takes for us to get the full length sequence that we would be comfortable with using. So, sometimes in an antibody, there might be a more hydrophobic area than normal. And if that happens, the bioinformatics team just says to the lab, hey, can you do an additional digestion with these different enzymes so we can get more peptides in this section? And that usually happens that same day or the day after. Things move pretty quickly over here because everyone is under one roof. And so, this last step is very crucial so that we're obtaining 100% accuracy and 100% coverage across the entire sequence. So getting started with REMAB is quite simple. All we need is 50 micrograms, so a really small amount of your protein. And since it's a completely de novo process, we can handle any species and any isotype. And we just ask that it's of reasonable purity, about 80% or more. And like I said, we are looking for a minimum coverage of 30 times depth of peptides across the whole sequence. Typically, we're getting hundreds of times coverage like you saw in that example I had and even on Shervin's presentation. And the samples are all independently reviewed for accuracy and also the isobaric checks. So this next section of my presentation, I'm just going to briefly go through a few other examples of how antibody sequencing has enabled antibody engineering with some other researchers that we've worked with as well. So this first example is from Dr. DeLuca at Colorado State University. And this lab studies mitosis and kinetochore misregulation. And they depend on antibodies for several different in vitro assays. They experienced inconsistent and poor performing antibodies from their reagent supplier, which made them pivot to do sequencing of their antibodies so that they can recombinantly express them to maintain consistent and reliable stocks of their reagents. And unfortunately, this is a pretty common thing that we see a lot from people. So we kind of always say, if you have your favorite antibody or something that you rely on so much, it's a really good idea to just get it sequenced to essentially digitalize that antibody in the event that it's discontinued or there's a mutation and it doesn't work anymore in your assay. So Dr. DeLuca's lab actually went on and did some engineering on the antibodies that they sequenced. And they were studying the HEC1 protein, which is a subunit of their kinetochore complex. And from obtaining the antibody sequence, they engineered various different formats, including fragment engineering, which was into SCFVCs, SCFVs, and Fab. And they use this for real-time antigen tracking for immunofluorescence, as well as other benefits of fragments. They also did species switching, which was used for immunofluorescence and the requirement of different antibody species specificity. And then also they did some reverse engineering of the antibody fragments into full-length bivalent antibodies. We also worked with Dr. Zubrev from the Karolinska Institute in Sweden, and this lab was studying isoaspartate accumulation in human blood serum albumin, and it was used as a biomarker for the detection of Alzheimer's disease. The blood biomarkers of Alzheimer's has gained a lot of attention, and it's allowed for earlier detection and diagnostics of this disease. And so with Dr. Zubrev's lab, their aim was to develop an ELISA that could measure isoasp levels in human plasma samples, which could supplement for diagnostics for Alzheimer's. Now, a set of isoaspartate-specific MABs were first generated in hybridomas, and we did de novo sequencing for them and identify the sequences of their candidate MABs. And we actually found a difference of 2 amino acids, as well as some glycan changes between a few of their antibodies, which resulted in actually a 3 orders of magnitude decrease in the specificity between their antibodies, which was interesting. That little change can make that big of a difference. And so, as a conclusion to this one, de novo sequencing enabled the lead selection of their candidate antibodies for further assay development and characterization. And lastly, the Vessler lab at the University of Washington helped make significant progress in developing antibody drugs targeting Nipah virus and Hendra virus in humans. They had previously generated 2 antibodies and needed to characterize them and their interactions with the virus. So they, to develop rational strategies for antibody engineering and drug design, a detailed understanding of the epitope recognition was needed, including the MAB sequences and the MAB glycoprotein structures. So this lab also did remab de novo sequencing with us to determine the amino acid sequence of their 2 candidate MABs. And using the sequence information, they humanized 12B2 and 1F5 by exchanging their constant heavy and light chain domain sequences with human and generated their own Fab fragments. And following this, they studied the 3D structure of the antigen and antibody complex to determine the value of using a cocktail of MABs to neutralize the virus. So in summary, we've talked a lot about remab today, which is used in many places across antibody development, a lot, of course, in target validation, and then also later on in lead profiling. And as you were introduced in the beginning of this webinar, we also offer a variety of other services, such as polyclonal sequencing, which can be used for either antibody discovery or keeping a supply of a polyclonal that you really love. So as we know, polyclonals tend to have a lot more variability than monoclonals might. So you can repab actually allows you to make a recombinant polyclonal from that serum sample. Additionally, for perhaps some later stage resources, we can use epitope mapping by HDXMS to really get an accurate and detailed view of where that exact binding region is between the antibody and the antigen. and also SPR to get an idea of those kinetic profiles. And that's it for me. So we're happy to enter the Q&A section now.
Great. No, thank you both for a great presentation. So the floor is open for any kind of questions. I don't see anything in the chat right now, but maybe we can just get started. I have a couple questions for you, Dr. Tabrizi. Thank you again for a really interesting presentation and a really cool application of antibodies to be able to improve the sensitivity of cell-free DNA. I was just wondering, from, I guess, the half-life perspective, since you are engineering the Fc domain of these antibody these DNA binding antibodies, do you see these priming agents persisting in your animal models for several months? And I was just wondering, like, could one priming event be able to protect the DNA for multiple collections over the course of several months?
Yeah, that's a great question. We've done some experiments to look at sort of the long-term persistence. We don't actually see a significant effect past a few days, in mice at least. But in principle, yeah, it's something that you could work on tuning to get the best sort of possible impact, and maybe a longer term impact if you can further enhance the circulation half-life of the DNA bound to the antibody. It always also becomes a balance, because I think to translate it to the clinic, I think people might feel a little more reassured about something that has a transient effect rather than a very long-term effect. And something that I didn't get too much into, but I think is important, is these antibodies, they also, like, DNA binding antibodies occur in diseases, like in some autoimmune diseases like lupus. Now those have intact Fc domains and have immunologic consequences that we can engineer out with our priming agents through silencing the Fc domain, but making sure that these are safe and tolerable in patients is also going to be critical. So I think that's a key parameter that will need to be optimized for clinical application.
Sounds good. And my other question also is, do you see that these priming agents would be suitable for just general use, or would you expect different priming agents to be created for different assays?
Yeah, that's a good question. For cell-free DNA, these should really work for any application where getting more cell-free DNA would be valuable, so that it should, in principle, apply to any tumor type, and maybe even in other applications like infectious disease, where sequencing and profiling circulating nucleic acids is a key diagnostic modality. I think in terms of like the actual clinical application that I foresee, I think the biggest immediate application would be for patients who already have a known cancer diagnosis and then have undergone through treatment. And basically the question is whether or not they have any residual disease in the body. And that's where exquisite sensitivity is really important, and we're still missing a lot of cases. And I think that's where these things could provide a needed boost to know for sure. But beyond that, you could also imagine applications in even early cancer detection, perhaps for patients who are at high risk of developing cancers, like smokers or other patient populations, where perhaps a liquid biopsy test using these priming agents could enable far earlier and more effective screening compared to what we have today.
No, that's great. Thank you for for those answers. It seems like we have a bit of a shy group today, but if there's no other questions, no, we will be sharing this recording with everyone on our website and through our LinkedIn. And if you have any other questions for Dr. Tabrizi or Jennifer, they have their emails here on the slide. So again, thank you so much to both of you for presenting another wonderful webinar. And Dr. Tabrizi, I wish you the best of luck with the next stages of this development. It's very exciting.
Thank you so much. Thanks for having me.
All right, we'll close the webinar here. Thank you, everyone. Have a nice day.
Thank you.